Many people think of dinosaurs as the dominant animals of the prehistoric world, but some of the most remarkable creatures lived after the dinosaurs disappeared. Millions of years ago, in the warm, swampy forests of what is now South America, an enormous snake called Titanoboa became one of the most powerful predators on Earth. Titanoboa was the largest snake ever discovered. Living about 58 million years ago, it thrived in a hot climate that helped reptiles reach much larger sizes than most can today. As a top predator, Titanoboa hunted a variety of animals and dominated the rivers and wetlands where it lived. How did this giant snake become so large? What did it eat, and why did it eventually disappear? $60 million years ago, in a time when the Earth was undergoing major changes in climate, land and living creatures, a very large prehistoric snake existed in what is now known as northern South America. During this period, the land that is now Colombia had a very warm and humid environment, with wide river systems, thick vegetation and swamp-like regions that supported many kinds of animals and plants. The remains of this snake were discovered much later, through scientific study of fossils preserved in layers of rock that formed in ancient river environments. These fossil remains showed that this snake had a long body structure supported by many vertebrae, and its bones were found in a region known for preserving plant fossils, fish remains, and other reptile traces from the same time period. The name given to this prehistoric snake is Titanoboa, a name that was created from scientific classification based on its fossil structure. The discovery of Titanoboa came from a fossil mining area in northern Colombia in a geological formation known as Terra Jaune. This formation contains layers of coal and sediment that were formed over millions of years from plant material and river deposits. The environment at the time when Titanoboa lived was filled with slow-moving water systems and dense plant growth along river banks. These conditions supported a wide range of life forms that depended on warm temperatures and steady humidity levels. The fossil evidence of Titanoboa includes many vertebrae that were found separated, but still preserved in good condition. These bones showed clear signs of belonging to a snake due to their shape, structure, and arrangement. Scientists carefully studied each fossil piece and compared them with modern snake skeletons in order to understand how the animal's body was formed. The vertebrae showed that the snake had a long and thick body that allowed it to move through water and land areas within its environment. The fossil remains were collected from rock layers that also contained fossils of ancient turtles, crocodile relatives, and large fish species that lived in the same time period. The Terra Jaune formation where Titanoboa was discovered is made up of layers of sediment that formed in a tropical swamp environment. These sediments were created by rivers that carried soil, plant material, and organic matter from surrounding areas and deposited them over time. As these layers built up, they preserved remains of plants and animals that lived in the region. The coal deposits found in this formation indicate that large amounts of plant material accumulated and were compressed over long periods of time. The presence of fossilized plant remains such as leaves, wood fragments, and seeds provides evidence of dense vegetation that covered the land surface. The study of Titanoboa fossils involved careful cleaning and preparation of the bones. Scientists removed surrounding rock material using fine tools and chemical processes to expose the structure of each vertebra. Once the bones were fully exposed, detailed measurements were taken to record their shape and size. These measurements were then compared with known snake species from modern times in order to understand how Titanoboa's body was arranged. The structure of the vertebrae indicated strong muscular attachment points, which suggest that the snake had a powerful body capable of movement through both water and land environments. The environment in which Titanoboa lived was influenced by high levels of heat and moisture. The region was located close to the equator, where sunlight exposure remained strong throughout the year. This led to stable warm conditions that supported continuous plant growth and active ecosystems. Rivers in this region moved slowly and often changed direction over time, creating wetlands and floodplains. These wet environments provided suitable living conditions for many reptiles, amphibians, fish, and plant life. The fossil record from this area shows that many species depended on water systems for survival and reproduction. Titanoboa's skeletal structure suggests that it belonged to a group of snakes known for having elongated bodies and flexible spines made up of many vertebrae. Each vertebra played a role in supporting movement and flexibility. The arrangement of these bones allowed the body to bend and stretch in different directions. The fossil vertebrae found in Serpent show that Titanoboa had thick bone structures, which indicate a strong body frame. The size and density of the bones suggest that the snake was adapted to an environment where movement through water was important for survival and hunting behavior. The discovery of Titanoboa also provided information about the types of animals that lived in the same ecosystem. Fossils of large reptiles with armored bodies were found in the same rock layers, indicating that waterways contained many types of predators and prey. Fish fossils from this period show that aquatic life was abundant, with species adapted to slow-moving rivers and swamp conditions. The presence of plant fossils indicates that the land surrounding these waterways was heavily covered with vegetation that provided shelter and food sources for many organisms. The process of identifying Titanoboa required collaboration among paleontologists, geologists, and fossil preparation specialists. Each group contributed knowledge about rock layers, fossil structure, and biological classification. The fossils were studied in laboratories where detailed analysis of bone microstructure was conducted. Microscopic examination of the vertebrae helped determine growth patterns and physical development of the snake over time. The internal structure of the bones showed signs of slow growth and steady development, which is consistent with reptiles that live in stable environments. The Serpent fossil site continues to be an important location for understanding prehistoric ecosystems. Layers of rock in this region provide a timeline of environmental changes that occurred over millions of years. Each layer contains different types of fossils that represent changes in plant and animal life. The preservation of Titanoboa fossils within these layers indicates that the snake lived during a period when conditions supported large reptile species and dense forest environments. The fossil record from this site also includes evidence of changing river systems that shaped the landscape over time. Titanoboa's body structure, based on fossil evidence, shows that the snake had a long spine supported by many vertebrae connected in a flexible arrangement. The skull remains have not been fully recovered, but related fossil fragments suggest that the head was designed for capturing and consuming large prey available in its environment. The jaw structure of related snake species shows that flexible joints allow for wide opening of the mouth, which is a feature common in large snake species. This anatomical feature would have been important for feeding in an environment with a variety of aquatic and semi-aquatic animals. The study of Titanoboa also provides insight into the temperature conditions of the ancient Earth during the time it lived. Fossil evidence from plants and reptiles suggests that the region maintained consistently warm conditions that supported reptile metabolism and growth. The presence of large reptiles in the fossil record is associated with environmental conditions that allowed cold-blooded animals to maintain active biological processes throughout the year. The warm and humid climate supported continuous biological activity without long periods of environmental stress caused by seasonal changes. The fossil vertebrae of Titanoboa were found in multiple locations within the Serpent Formation, indicating that remains of the species were distributed across different parts of the ancient river system. Each fossil discovery added more information about the structure and distribution of the species within its environment. The arrangement of fossils within sediment layers suggests that the remains were carried by water movement after death and later buried by sediment deposits. Over time, these remains were preserved through mineralization processes that replaced organic material with minerals from surrounding sediments. The research process also involved reconstructing the likely body proportions of Titanoboa based on vertebrae size and spacing. Each vertebra provides information about the length and thickness of different sections of the body. By analyzing these patterns, scientists were able to estimate the general structure of the snake's body. The spacing of vertebrae suggests that the body had consistent thickness along much of its length, with variations in structure near the head and tail regions. These anatomical features provide important information about movement and lifestyle. The surrounding fossil evidence from the same geological formation includes remains of ancient plant species, such as large-leafed tropical trees, vines, and aquatic vegetation. These plants grew along riverbanks and in swampy areas where water levels fluctuated throughout the year. The plant fossils show that the environment had a dense coverage of vegetation, that it played a role in stabilizing soil and supporting diverse animal life. The interaction between plant life and water systems created conditions that allowed for a complex ecosystem to exist. The study of Titanoboa continues through ongoing fossil research, where new fragments may still be discovered in sediment layers of the Serpent Formation. Each new fossil piece contributes additional information about the structure and lifestyle of this prehistoric snake. The careful documentation of each discovery helps build a clearer understanding of how the species lived and interacted with its environment. Fossil preparation techniques continue to improve, allowing for more detailed analysis of bone structure and composition. Titanoboa remains an important subject in the study of prehistoric reptiles and ancient ecosystems. Its fossil record provides evidence of a time when Earth's tropical regions supported large reptile species in warm and humid environments. The geological and biological information preserved in the Serpent Formation continues to offer valuable insight into ancient river systems, plant life, and animal diversity during this period. The combination of fossil evidence and geological study helps build a detailed picture of environmental conditions that existed during the time Titanoboa lived. The structure of Titanoboa fossils also provides information about how sedimentation processes worked in ancient river environments. Layers of mud, sand, and organic material accumulated gradually over time, covering biological remains and protecting them from decay. Pressure from overlying sediments contributed to the preservation of fossil structures. Mineral-rich water passing through the sediment layers slowly replaced organic materials with stone-like substances, preserving the shape of bones and plant matter. This process allowed scientists millions of years later to recover detailed fossil evidence from deep within the rock layers. Further analysis of Titanoboa fossils shows that different parts of the snake's body were preserved in separate locations, suggesting movement of remains by water currents before burial. The distribution of fossils across the site indicates that river systems played a role in transporting biological material after death. This movement of remains before fossilization is a common feature in river-based fossil sites. After the extinction of the dinosaurs, the Earth entered a long period of change, where life slowly adjusted to new conditions, and many new forms of living things began to appear across land, water, and air. This time began about 66 million years ago, when a major extinction event ended the dominance of large dinosaurs and removed many species that had lived on Earth for millions of years before that moment. The environment after this event was very different from what existed during the time of dinosaurs. Large areas of land were changed, many plant types were affected, and the balance of ecosystems shifted in ways that allowed smaller surviving animals and plants to expand into new roles. In the earliest period after the extinction, many regions of Earth were left with fewer large animals. This created space for surviving species to spread into areas that were previously occupied. Small mammals, birds, reptiles, amphibians, and insects were among the groups that continued living through the extinction event. These surviving groups had to adjust to changes in food sources, temperature patterns, and available habitats. Over time, these adjustments led to the development of new species that slowly filled the ecological roles left empty. Plant life also went through major changes during this time. Many plant species that had depended on interactions with dinosaurs were affected, while other plants adapted to new conditions and spread widely. Forests began to change in structure, with flowering plants becoming more common in many regions. These plants produced flowers and fruits that supported new forms of insect life and helped create new feeding relationships between species. Seeds spread through wind, water, and animals, allowing vegetation to expand into areas that had been disturbed. The climate during the early period after the extinction changed gradually. Some regions experienced warmer conditions, while others experienced cooler phases, depending on atmospheric changes and the movement of landmasses. These climate shifts influenced how plants and animals survived and where they could live. In many areas, ecosystems were still unstable, and species had to continue adapting in order to survive long-term changes in temperature, rainfall, and seasonal patterns. Mammals began to grow in diversity after the extinction event. Before this time, mammals existed, but were generally small and lived in limited ecological roles. After the disappearance of dinosaurs, mammals began to spread into many different environments. Some lived in forests, some in open land, and others near water sources. Their diets also became more varied, including plants, insects, and other small animals. Over many millions of years, mammals developed a wide range of body structures and survival strategies, allowing them to occupy many ecological roles across the planet. Birds also expanded during this period. Birds are closely related to certain groups of dinosaurs that survived the extinction event. After the extinction, birds developed into many different forms and adapted to various environments. Some became strong flyers that traveled long distances, while others became ground-dwelling species that lived in forests or open areas. Their beaks, wings, and feeding habits changed gradually over time as they adapted to different food sources such as seeds, insects, fish, and plants. Reptiles continued to survive after the extinction and remained important in many ecosystems. Some reptile groups expanded into new habitats, including freshwater environments and dry land areas. Crocodile-like species continued to live in rivers and lakes, feeding on fish and other animals. Lizard and snake groups also increased in variety, adapting to different climates and ecological conditions. In marine environments, reptiles had already been present before the extinction event, and some groups continued to survive afterward, adapting to ocean life in different ways. Insects experienced major expansion during the period after the extinction. Because insects are small and reproduce quickly, they were able to adapt to changing plant life and environmental conditions. As flowering plants became more widespread, insects developed new relationships with them. Many insects began feeding on nectar, pollen, leaves, and plant sap. Some insects also became important in spreading plant pollen, helping plants reproduce and spread across large areas of land. This interaction between insects and plants became an important part of many ecosystems. Marine life also changed significantly after the extinction event. Many ocean species disappeared, but surviving species adapted to new conditions in the seas. Fish populations diversified over time, and new types of marine predators and smaller marine organisms appeared. Coral systems developed in different ways, depending on water temperature, depth, and nutrient availability. Ocean ecosystems slowly rebuilt themselves. As surviving organisms expanded, and new species formed over millions of years. Rivers and lakes also became important environments for developing life. Freshwater ecosystems supported a wide range of organisms, including fish, amphibians, insects, and plants. These environments often changed due to shifting rainfall patterns and geological changes that affected water flow. Species living in freshwater environments adapted to seasonal changes and varying water levels, which influenced how they reproduced, fed, and moved within their habitats. Landscapes after the extinction were also shaped by geological activity. Movement of Earth's surface continued over long periods of time, forming mountains, valleys, plains, and other land structures. These changes affected climate patterns and created different environments, where species could evolve separately. As land masses shifted, populations of animals and plants became separated from each other, allowing them to develop different traits over time depending on local conditions. Soil development played an important role in supporting new plant life. As plants grew and decayed, they contributed organic material to the ground, which improved soil quality in many regions. This process allowed forests and grass-like plant systems to spread in different areas. The availability of nutrients in soil influenced which plants could grow in specific regions, and this in turn affected which animals could live there. Over time, ecosystems became more complex as more species appeared and interactions between them increased. Food chains developed where plants supported herbivores, herbivores supported carnivores, and decomposers broke down dead material to return nutrients to the soil. These relationships formed across land, water, and air environments, creating interconnected systems that continued to change as new species evolved. Temperature and atmospheric composition continued to shift over long periods after the extinction. Levels of gases in the atmosphere influenced climate conditions, which affected plant growth and animal survival. Some periods had higher temperatures that supported dense vegetation in certain regions, while other periods had cooler conditions that changed the types of plants that could survive. These changes influenced how ecosystems developed across different parts of the planet. The movement of continents over millions of years also played a major role in shaping life after the extinction. As landmasses slowly changed position, climates in different regions were altered. Some areas became wetter, while others became drier. These environmental differences encouraged the development of species that were adapted to specific conditions. Isolation of populations in different regions allowed genetic changes to accumulate over time, resulting in new species emerging in separate locations. During this long period, many early forms of modern animal groups began to appear. Early relatives of large mammals, marine mammals, and flying mammals developed gradually from smaller ancestors. These animals adapted to specific environments such as oceans, forests, and open plains. Their body structures changed over time in response to food availability, movement patterns, and environmental conditions. Plant evolution continued steadily after the extinction. Flowering plants expanded into many regions, producing a wide variety of forms, sizes, and reproductive methods. Some plants developed deep root systems to access water in dry environments, while others adapted to wet environments with frequent flooding. The diversity of plant life supported increasing diversity in animal life, as animals depended on plants for food and shelter. Insects developed more specialized roles in ecosystems. Some insects became highly adapted to specific plants, feeding only on certain types of leaves or flowers. Others developed behaviors that allowed them to live in soil, water, or inside plant structures. This specialization increased diversity among insect groups and influenced how plants and insects interacted over time. As time continued, ecosystems became more stable in many regions. Species interactions became more established, and energy flow through ecosystems became more consistent. However, environmental changes still occurred due to volcanic activity, climate shifts, and geological movement. These changes occasionally affected ecosystems, requiring species to adapt or move to new areas. The development of new habitats continued as landscapes changed. River systems shifted paths, coastlines changed shape, and forests expanded or contracted depending on climate conditions. These changes created new environments where species could develop different survival strategies. Over long periods, life became more widely distributed across the planet, reaching many different ecological zones. The early period after the extinction also saw changes in oxygen levels in some environments. These changes affected how animals and plants functioned, including their growth rates and energy use. Species adapted gradually to these conditions through biological adjustments that occurred over many generations. The oceans continued to develop new ecosystems as coral systems, plankton populations, and fish communities evolved. Marine food systems became more complex, with many layers of interaction between small organisms and larger predators. Ocean currents influenced nutrient distribution, which affected where marine life could thrive. Freshwater systems also supported increasing diversity. Lakes and rivers became environments where fish and amphibians developed specialized traits for survival in changing water conditions. Some species adapted to fast-moving waters, while others adapted to still water environments. Seasonal changes influenced reproduction cycles and feeding patterns in many freshwater species. Over millions of years after the extinction, the Earth developed a wide range of environments supporting diverse life forms. These environments included dense forests, open land areas, freshwater systems, coastal regions, and deep ocean habitats. Each environment supported different combinations of plants and animals that adapted to local conditions and changing climates. The long period after the extinction event was marked by continuous biological development and environmental change. Life gradually expanded into many new directions, forming complex systems of interaction between species and their surroundings. About 60 million years ago, during the early part of the Paleocene epoch, a giant snake moved through the hot and wet forests of what is now northern South America. This enormous reptile lived in a world that was still recovering from the mass extinction that had ended the age of the non-avian dinosaurs. For millions of years, its existence remained completely unknown to human beings. Its bones lay buried beneath layers of rock and sediment deep underground. Rain fell, rivers changed course, mountains slowly rose, and entire ecosystems appeared and disappeared while the remains of this giant animal stayed hidden from view. The discovery of Titanoboa fossils in Colombia became one of the most important paleontological findings of the modern era. The fossils revealed the existence of the largest snake ever known to science and opened a new window into understanding life in the ancient tropical environments that existed after the extinction of the dinosaurs. The discovery not only introduced scientists to a remarkable animal, but also provided valuable information about the climate, plants, animals, and ecosystems of the ancient world. The story began in northern Colombia, in a region known for rich coal deposits. Coal mines often contain ancient rock layers that preserve fossils, because the environments in which coal forms are usually filled with plant material and sediments capable of burying dead organisms. These sediments can protect bones, leaves, shells, and other remains for millions of years. One of the most important locations connected with the discovery was the Cerrejón coal mine in the La Guajira region of Colombia. This mine is among the largest open-pit coal mines in the world. The area contains rock formations that date back to the Paleocene epoch, a period that began shortly after the extinction event that ended the reign of the dinosaurs. Scientists had long known that the Cerrejón formation contained ancient plant fossils and other remains, but the full significance of the site was not immediately understood. As mining operations removed large amounts of rock and earth, ancient layers became exposed. These newly revealed rock formations provided scientists with rare opportunities to study sediments that had once formed part of a tropical rainforest environment. Researchers visiting the mine noticed that the exposed rocks contained a wide variety of fossils. These remains included plants, turtles, crocodile relatives, fish, and other organisms that had lived millions of years earlier. During the early years of investigation, teams of scientists began collecting fossils from different sections of the mine. Their goal was to understand the ecosystems that existed in northern South America shortly after the extinction of the dinosaurs. The fossils they discovered painted a picture of a lush tropical environment filled with rivers, swamps, dense vegetation, and abundant wildlife. Among the many fossils being collected were unusual vertebrae. Vertebrae are the bones that make up the backbone of animals. At first glance, these bones appeared to belong to a snake. However, the size of the vertebrae immediately attracted attention. They were far larger than those of any modern snake known to the researchers. The fossil vertebrae were recovered from rock layers dating to approximately 60 million years ago. Scientists carefully examined the bones and compared them with skeletons of living snakes preserved in museum collections. The more they studied the fossils, the more extraordinary the discovery appeared. The vertebrae displayed features that clearly identified them as belonging to a member of the boa family. Modern boas include large snakes, such as the boa constrictor and the anaconda. The fossil bones shared important anatomical characteristics with these living relatives, yet the fossil vertebrae were much larger than those found in any known living species. Researchers realized that they were dealing with an animal unlike anything previously discovered. More fieldwork followed as scientists searched for additional fossils. Over time, numerous vertebrae and rib bones were recovered from the Cerrejón formation. These remains came from multiple individuals rather than a single snake. The discovery of fossils from several animals helped confirm that the giant snake was not merely an unusually large member of a known species. Excavating fossils in a coal mine presented both opportunities and challenges. The exposed rock layers allowed researchers to reach fossils that would otherwise remain buried underground. However, mining activities could also damage fossil-bearing rocks if specimens were not identified and collected in time. Scientists therefore worked carefully and often collaborated with mine personnel to locate promising fossil sites. Each fossil discovery required detailed documentation. Researchers recorded the precise location of every specimen, noted the surrounding rock layers, and examined the geological context in which the bones were found. Such information was essential for determining the age of the fossils and understanding the ancient environment in which the animals had lived. The fossils were transported to laboratories where they underwent cleaning and preparation. Sediment attached to the bones had to be removed carefully using specialized tools. Even though fossilized bones are often hard, they can still be fragile. Technicians spent many hours exposing important anatomical details while ensuring that the specimens remained intact. As the collection of giant snake fossils grew, scientists began conducting detailed measurements. They compared the vertebrae with those of modern boas and anacondas. In snakes, vertebral size is closely related to overall body length. By examining the relationship between vertebral dimensions and body size in living species, researchers could estimate the length of the ancient snake. The results were astonishing. Calculations suggested that the giant snake measured roughly 13 meters in length. Some estimates placed its weight at more than 1 metric ton. Such dimensions exceeded those of any living snake. The animal represented a level of size never before documented in the fossil record. For snakes, the discovery required the creation of a new scientific name. Researchers named the giant snake Titanoboa kerejonensis. The name Titanoboa combines the word titan, referring to enormous size, with boa, indicating its relationship to modern boas. The species name Cerejonensis honored the Cerejon region where the fossils were discovered. The formal scientific description of Titanoboa attracted worldwide attention. News reports, documentaries, and scientific publications highlighted the significance of the discovery. People were fascinated by the idea that a snake of such immense size had once inhabited tropical forests in South America. The fossils also provided important clues about ancient climate conditions. Snakes are ectothermic animals, meaning that their body temperature depends heavily on the surrounding environment. Because large snakes require substantial warmth to maintain their biological processes, scientists realized that Titanoboa's enormous size could reveal information about temperatures in the Paleocene tropics. Researchers studied the relationship between body size and climate in living reptiles. Their analyses suggested that the tropical environment in which Titanoboa lived was warmer than many present-day tropical regions. This conclusion generated significant scientific discussion and encouraged further investigations into ancient climate systems. The Cerejon Formation yielded much more than snake fossils. Numerous plant remains were discovered alongside Titanoboa. Fossil leaves, seeds, fruits, and wood provided evidence of a rich rainforest ecosystem. Scientists identified many plant groups that continue to exist today, indicating deep evolutionary roots for tropical forest communities in South America. The fossil plants suggested dense vegetation supported by abundant rainfall and warm temperatures. Rivers and wetlands likely spread throughout the region, creating habitats suitable for large aquatic and semi-aquatic animals. Such environments would have provided ideal conditions for a giant snake. The discovery of fossil turtles further enriched understanding of the ecosystem. Several turtle species recovered from the site were exceptionally large. Their remains suggested that the environmental conditions could support reptiles of remarkable size. Some turtles possessed shells much larger than those seen in most living species. Fossils of crocodile relatives were also common. These predators inhabited rivers and wetlands and likely shared habitats with Titanoboa. Their presence demonstrated that the ecosystem supported numerous large carnivorous reptiles. The interactions between these animals remain a subject of scientific investigation. Fish fossils revealed the presence of productive aquatic environments. Rivers and swamp systems likely contained diverse fish populations that served as food for larger predators. The abundance of aquatic life would have supported complex food webs throughout the region. Scientists believe Titanoboa probably spent much of its time in or near water. This interpretation is based partly on its anatomical relationship with modern anacondas, which are highly adapted to aquatic habitats. Water could help support the immense weight of such a large snake and assist in movement. The fossil vertebrae themselves provided valuable anatomical information. Snake vertebrae contain numerous features that help scientists identify species and determine evolutionary relationships. Detailed examination revealed characteristics consistent with primitive boa lineages, while also displaying unique traits that distinguish Titanoboa from known species. The discovery helped fill an important gap in the fossil record of snakes. Although modern snakes are widespread and diverse, fossil evidence of their early evolution is often incomplete. Titanoboa provided direct evidence that giant boas existed relatively soon after the extinction of the dinosaurs. Researchers also investigated the geological history of the Serpent region. During the Paleocene, northern South America looked different from today. River systems, coastlines, and landscapes were shaped by geological processes that had been occurring for millions of years. Understanding these ancient environments helped scientists reconstruct the world in which Titanoboa lived. Sedimentary rocks preserved evidence of repeated flooding events. Layers of mud, sand, and organic material accumulated over long periods. These sediments eventually buried the remains of plants and animals, creating conditions favorable for fossil preservation. The process through which Titanoboa became fossilized began shortly after death. When an animal died in a river or swamp environment, sediments could rapidly cover the body. Burial protected bones from scavengers and reduced exposure to weathering. Over millions of years, minerals replaced organic materials, transforming the bones into fossils. Not every part of a skeleton survives fossilization. Many factors influence preservation, including environmental conditions, burial rate, and geological activity. The fact that numerous Titanoboa vertebrae were recovered indicates that at least some individuals experienced favorable preservation conditions. Field expeditions to Serpent continued after the initial discovery. Scientists searched for additional fossils that might provide more information about Titanoboa's anatomy and biology. Each new specimen contributed to a growing understanding of the species. Researchers used advanced imaging techniques to study the fossils. High-resolution photography, digital measurements, and computerized analyses allowed scientists to examine details that might otherwise be difficult to observe. These methods improved the accuracy of anatomical descriptions and size estimates. The discovery also stimulated interest in South American paleontology. Many fossil sites in the region remain relatively unexplored compared with some areas of North America and Europe. Titanoboa demonstrated the scientific importance of tropical fossil deposits and encouraged additional research projects. Educational institutions around the world incorporated Titanoboa into discussions of evolution, climate change, and prehistoric life. museums created exhibits featuring reconstructions of the giant snake. Visitors could see models illustrating its estimated size and learn about the ancient rainforest environment in which it lived. Creating accurate reconstructions required careful scientific work. Artists collaborated with paleontologists to ensure that body proportions, coloration assumptions, and environmental settings reflected available evidence. Although certain details remain uncertain, the reconstructions helped communicate the significance of the discovery to the public. The naming and description of Titanoboa represented the efforts of many scientists from different fields. Paleontologists, geologists, botanists, climate researchers, and fossil preparation specialists all contributed important expertise. Such interdisciplinary collaboration was essential for understanding the broader significance of the fossils. The discovery highlighted the value of coal mine fossil sites. Industrial activities sometimes expose geological formations that would otherwise remain inaccessible. When scientists and industry cooperate, important fossil discoveries can be documented and preserved before they are lost. The giant snake fossils also encouraged researchers to investigate the ecological consequences of the dinosaur extinction. The disappearance of large dinosaur species created opportunities for other groups of animals. During the Paleocene, mammals, birds, reptiles, and other organisms diversified in changing ecosystems. Titanoboa demonstrated that reptiles remained important components of these ecosystems. The snake occupied a position among the largest predators in its environment. Its existence revealed that giant reptiles continued to thrive millions of years after the extinction event that ended the age of the non-avian dinosaurs. The fossil record from Serpent continues to provide new insights. Additional discoveries have expanded knowledge of Paleocene biodiversity and environmental conditions. Every fossil contributes another piece of information about life in ancient, tropical South America. Scientists continue studying Titanoboa because many questions remain unanswered. Researchers seek to understand its growth rate, reproductive biology, hunting behavior, and ecological role. Fossils alone cannot answer every question, but each new discovery adds valuable evidence. The discovery also influenced studies of snake evolution. By examining anatomical features preserved in Titanoboa, scientists can better understand relationships among ancient and modern boas. These analyses help trace evolutionary changes across millions of years. Public fascination with Titanoboa has remained strong since the fossils were first announced. The idea of a snake reaching such extraordinary dimensions captures attention and sparks curiosity about prehistoric life. Educational programs frequently use Titanoboa to introduce audiences to paleontology and geological history. The Serpent fossils serve as reminders of how much remains hidden within Earth's rocks. Vast numbers of organisms lived and died over geological time, leaving traces that may still await discovery. Every fossil site has the potential to reveal species previously unknown to science. For Colombia, the discovery brought international recognition to the country's paleontological resources. Researchers from around the world became interested in studying the region's ancient rock formations. Collaborative projects expanded scientific knowledge and increased awareness of Colombia's geological heritage. The discovery of Titanoboa fossils transformed understanding of Paleocene ecosystems. What began as the recovery of unusual vertebrae from a Colombian coal mine developed into one of the most significant fossil discoveries of the 21st century. Through careful excavation, analysis, and interpretation, scientists uncovered evidence of a giant snake that lived about 60 million years ago in a warm tropical rainforest filled with diverse plants and animals. The fossils recovered from the Serpent Formation continue to provide information about ancient climates, ecosystems, evolutionary history, and the remarkable reptile known as Titanoboa. The size and weight of Titanoboa became one of the most fascinating aspects of the discovery. Scientists carefully examined fossilized vertebrae, ribs, and other skeletal remains recovered from ancient rock layers in Colombia. These remains showed that Titanoboa was not simply a large snake. It was an animal of exceptional proportions that reached lengths and body masses that few other reptiles have ever achieved. Estimating the exact size of an extinct animal is not always easy. Fossils rarely preserve an entire skeleton. In most cases, scientists must work with incomplete remains and use measurements from living relatives to estimate the dimensions of extinct species. In the case of Titanoboa, researchers compared the fossil vertebrae with those of modern boas and anacondas. The vertebrae of snakes vary in size according to the length and thickness of the body. By examining these relationships, scientists were able to estimate Titanoboa's overall dimensions with reasonable confidence. The results were remarkable. Titanoboa is believed to have reached a length of around 42 to 47 feet. Some estimates suggest that certain individuals may have been even longer. A snake of such length would have stretched across a large area when fully extended. Its body would have occupied a significant amount of space, making it one of the most impressive reptiles ever discovered in the fossil record. Length alone does not fully describe the immense size of Titanoboa. Equally important was its thickness. Modern giant snakes such as green anacondas have very muscular bodies, but Titanoboa appears to have been much thicker. Fossil evidence suggests that the body of this giant snake was exceptionally wide. In some areas, its diameter may have approached 3 feet. This immense girth contributed greatly to its overall weight and strength. Weight estimates for Titanoboa indicate that it may have weighed approximately 1 1/4 tons. Some calculations suggest a body mass of more than 2,500 pounds. This weight placed Titanoboa among the heaviest snakes ever known. Carrying such enormous mass required a powerful skeletal structure and massive muscles distributed throughout its body. The vertebrae of Titanoboa provide valuable clues about its weight-bearing abilities. Snakes rely on hundreds of interconnected vertebrae to support movement and maintain body stability. In Titanoboa, these vertebrae were exceptionally large and robust. Their size suggests that they supported a body carrying tremendous mass. Each vertebra formed part of a flexible yet strong framework that allowed the snake to move while supporting its immense weight. The muscles attached to these vertebrae were likely very powerful. Snakes use muscles throughout their bodies to move, climb, swim, and capture prey. Titanoboa's enormous dimensions meant that its muscles had to generate significant force. Every movement would have involved the coordinated action of thousands of muscle fibers working together. This muscular system helped the snake propel itself through water, maneuver through wetlands, and overpower prey animals. Scientists believe that Titanoboa spent much of its life in and around water. An aquatic lifestyle may have helped support its tremendous weight. Water provides buoyancy, reducing the strain that gravity places on large animals. By spending significant time in rivers, swamps, and wetlands, Titanoboa could move more efficiently than it would have on dry land. Its massive body would have been easier to support in water, allowing it to conserve energy while remaining an effective predator. The warm climate of the Paleocene played an important role in allowing Titanoboa to achieve such extraordinary size. Snakes are ectothermic animals, meaning that they depend largely on external sources of heat to regulate body temperature. Larger snakes require substantial warmth from their environment to maintain normal biological functions. The tropical regions where Titanoboa lived were significantly warmer than many modern tropical environments. These conditions may have provided the heat necessary to support the metabolism of such a gigantic reptile. Researchers have used Titanoboa's size to estimate ancient temperatures. The reasoning is based on the relationship between body size and environmental temperature in modern reptiles. Since Titanoboa grew much larger than any living snake, the environment in which it lived must have provided sufficient warmth to sustain such growth. Studies suggest that average temperatures in its habitat were likely higher than those found in many present-day tropical forests. Growth to such enormous dimensions would have required a steady and abundant food supply. Titanoboa lived in ecosystems that recovered after the extinction of the non-avian dinosaurs. During this period, tropical forests expanded and supported a wide variety of animals. Rivers and wetlands contained numerous fish, reptiles, and other creatures that could serve as food. Access to plentiful prey allowed Titanoboa to obtain the energy necessary for growth and maintenance. As Titanoboa increased in size during its lifetime, its nutritional requirements would also have increased. Young individuals likely fed on smaller prey before gradually shifting to larger animals as they matured. Each stage of growth required increasing amounts of food. Over many years, consistent access to prey enabled the snake to reach extraordinary dimensions. The relationship between length and weight in Titanoboa is particularly interesting. Weight does not increase at the same rate as length. As an animal becomes longer, its volume and mass increase much more rapidly. This means that even a modest increase in length can result in a substantial increase in weight. Because Titanoboa combined exceptional length with exceptional thickness, its body mass reached extraordinary levels. A large portion of Titanoboa's weight came from muscle tissue. Snakes depend heavily on muscular strength because they lack limbs. Every action, from movement to feeding, relies on the coordinated contraction of muscles throughout the body. Titanoboa's enormous musculature enabled it to generate tremendous force when capturing prey. The skin of Titanoboa would also have contributed to its size. Large snakes possess thick scales and substantial skin layers that protect the body from injury and environmental hazards. Although skin does not account for most of the weight, the sheer dimensions of Titanoboa meant that its outer covering represented a considerable amount of tissue. The internal organs of Titanoboa were likewise enormous. Its heart, lungs, digestive system, and other organs had to support a body of unprecedented size. A giant snake requires efficient circulation to deliver oxygen and nutrients throughout its body. The heart would have pumped blood across an extensive network of vessels reaching from head to tail. Its lungs were probably exceptionally long. Modern snakes often possess elongated lungs that extend through much of the body cavity. In Titanoboa, these organs would have been enlarged to accommodate the demands of its massive body. Efficient respiration was essential for sustaining muscular activity and maintaining bodily functions. The digestive system of Titanoboa was capable of processing large meals. Snakes are known for swallowing prey whole. Once consumed, prey undergoes digestion over extended periods. A snake of Titanoboa's size could likely consume very large animals in a single feeding event. Such meals would provide substantial energy that could sustain the snake for long intervals. The head of Titanoboa, although not preserved completely in the fossil record, is believed to have been proportionally large. A larger head would allow the snake to consume bigger prey items. The jaws of snakes are highly flexible, enabling them to open widely during feeding. Titanoboa's jaw structure would have supported the capture and swallowing of sizable animals. Its neck region was also heavily built. The muscles surrounding the head and neck played important roles in prey capture and feeding. Strong neck muscles enabled the snake to control struggling prey and position it for swallowing. These muscles contributed further to the overall mass of the animal. The sheer weight of Titanoboa influenced the way it moved. Large snakes generally move more slowly than smaller species because of the energy required to shift their mass. Titanoboa's movements on land were likely deliberate and controlled. In water, however, its massive body could have moved with greater ease. Powerful lateral undulations would have propelled it through rivers and wetlands. The distribution of weight along Titanoboa's body was another important factor. Rather than concentrating mass in one region, the snake's weight was spread across its entire length. This distribution helped support stability and flexibility. Hundreds of vertebrae and associated muscles worked together to manage the enormous body mass efficiently. Scientists often discuss Titanoboa in relation to the limits of snake growth. Modern snakes face biological constraints that influence their maximum size. Factors such as climate, food availability, metabolism, and reproduction affect growth potential. Titanoboa demonstrates that under favorable environmental conditions, snakes could achieve dimensions far beyond those seen today. The fossil vertebrae discovered in Colombia remain among the strongest pieces of evidence for Titanoboa's immense size. These bones are noticeably larger than those of living giant snakes. Detailed measurements revealed dimensions consistent with a truly gigantic animal. The fossils allowed researchers to reconstruct body proportions and estimate mass using scientific methods. Each vertebra tells part of the story. The width, height, and shape of the bones reveal information about muscle attachments and body structure. Larger vertebrae indicate larger muscles and a larger overall body. Through careful analysis, scientists pieced together an image of a snake whose size exceeded previous expectations. The discovery attracted global attention because giant animals often capture public imagination. Titanoboa's dimensions challenged assumptions about the upper limits of snake size. Before its discovery, many people believed that modern anacondas represented the maximum size that snakes could achieve. Titanoboa demonstrated that ancient environmental conditions allowed even greater growth. The wetlands where Titanoboa lived provided ideal conditions for supporting such a massive reptile. Warm temperatures encouraged biological activity, while abundant water helped regulate body temperature and support movement. Rich ecosystems supplied food resources necessary for sustaining large predators. These factors work together to create an environment capable of supporting Titanoboa's exceptional size and weight. Reproduction in a snake of this magnitude would also have involved significant energy demands. Producing offspring requires substantial biological investment. Large females may have carried considerable numbers of developing young or eggs, depending on their reproductive strategy. Supporting reproduction would have required additional food intake and energy reserves. The growth rate of Titanoboa remains uncertain, but scientists believe that reaching full size likely took many years. Growth in reptiles often continues throughout life, although it slows with age. A young Titanoboa would gradually accumulate mass and length as it matured. Over time, small increases would have produced a truly enormous adult. Environmental stability was important for maintaining such growth. Long periods of favorable climate and abundant prey allowed individuals to continue developing. Interruptions in food availability or major environmental changes could have limited growth potential. The ecosystems of Paleocene South America appear to have provided conditions that supported giant reptiles for extended periods. The immense size of Titanoboa also influenced its ecological role. Large predators occupy important positions within ecosystems. Their feeding activities affect populations of prey animals and contribute to ecological balance. Titanoboa's extraordinary dimensions allowed it to exploit food resources unavailable to smaller predators. Its weight and strength gave it access to large prey items. Powerful muscles enabled it to restrain animals that would have been beyond the capabilities of smaller snakes. The combination of mass, length, and muscular force made Titanoboa one of the dominant predators in its environment. Scientists continue to study Titanoboa because it offers valuable insights into ancient climates, ecosystems, and reptile evolution. Every new fossil provides additional information about its anatomy and lifestyle. Research helps refine estimates of length, weight, and body proportions, while improving understanding of the environmental factors that supported such a giant snake. Even today, the estimated weight of approximately 1 1/4 tons remains astonishing. Few snakes in Earth's history are known to have approached this mass. The immense body required vast amounts of energy, strong skeletal support, and highly developed musculature. Every aspect of Titanoboa's biology reflected the demands imposed by its extraordinary size. The relationship between size and survival is evident throughout the anatomy of Titanoboa. Its vertebrae, muscles, organs, and feeding adaptations all reveal an animal built to function at a scale rarely seen among snakes. Each feature contributed to the successful operation of a body weighing thousands of pounds and extending across dozens of feet. As more fossils are examined, scientists continue refining their understanding of Titanoboa's dimensions. Different methods of calculation may produce slightly different estimates, but all indicate a snake of truly enormous proportions. Whether measured by length, thickness, or mass, Titanoboa stands among the largest snakes known from the fossil record. Its body represented millions of years of evolutionary adaptation within warm tropical ecosystems. The fossil evidence reveals an animal capable of reaching lengths exceeding 40 feet and weights surpassing 2,000 pounds. These dimensions highlight the remarkable biological possibilities that existed in Earth's ancient tropical environments, where abundant resources, warm temperatures, extensive wetlands, and stable ecological conditions supported the growth of one of the most massive snakes ever known. Titanoboa is placed within a larger biological group known as Boidae, which is a family of snakes that includes many forms of constricting snakes. These snakes belong to a long history of reptile evolution that developed during the time period known as the Late Cretaceous, when the land that is now called the Americas had different environments and living conditions compared to present times. Members of Boidae are part of a broad group of snakes that do not rely on venom for hunting, but instead depend on powerful body movement to hold and subdue their prey. The group has been recorded in many parts of the world, and it includes several subdivisions known as subfamilies, with a total count reaching 6 recognized subfamilies. These subfamilies are spread across different regions of the world, showing that Boid snakes have a wide geographical presence that covers nearly all continents, where suitable environments existed at different times in Earth's history. Within this family, Titanoboa is classified under the subfamily Boinae, based on details observed in its vertebral structure. The vertebrae are the bones that make up the backbone, and in snakes these bones carry important information used by scientists to determine relationships between extinct and living species. The structure, size, and arrangement of these bones provide clues about how the animal moved and how it was related to other snakes. The Boinae subfamily includes snakes that are primarily associated with regions in the Americas. These snakes are found in a range that extends through parts of North America and South America. Their distribution includes areas reaching as far north as what is now called Mexico, and also extends into island regions of the Caribbean, known historically as the Antilles. In the southern direction, members of this subfamily extend into parts of South America, reaching regions that include present-day Argentina. This distribution pattern reflects the presence of suitable environments across these regions at different points in geological history, allowing these snakes to live and diversify in many habitats. Titanoboa is recognized as part of this subfamily based on fossil evidence that has been studied in detail. Among the fossil materials that provided important information, skull fragments played a central role in confirming its placement within Boinae. The skull remains included structures that showed specific features related to the internal passageways of the skull. One of these features involved the reduced form of palatine choanals. The palatine choanals are openings within the skull that are involved in the passage of air and are positioned in a way that helps define relationships among snake groups. In Titanoboa, the reduced condition of these openings provided important information used in classification. The presence of this reduced structure matched patterns observed in other members of Boinae, which supported the interpretation that Titanoboa belongs within this group. The skull evidence added to the information already gathered from vertebrae, forming a more complete understanding of the anatomical structure of the animal. Research work that was presented in the year 2013 introduced additional interpretations of Titanoboa's relationships within the broader group of boid snakes. In that work, analyses based on available fossil characteristics placed Titanoboa in a position that showed close relation to certain snake taxa, found in regions that include Pacific Islands and Madagascar. These regions are located in different parts of the world, and the taxa found there represent groups of snakes that developed in isolated environments. The analysis used comparisons of anatomical features that are preserved in fossils, focusing on structural details that remain consistent over long periods of evolutionary change. The results of this research indicated that Titanoboa shared a connection with these groups in a way that provided insight into how different lineages of boid snakes are related across broad geographic regions. The interpretation from the 2013 research contributed to the understanding of how snake lineages that exist in different parts of the world may have developed from earlier ancestral groups. The relationship identified between Titanoboa and snake taxa from the Pacific Islands and Madagascar provided a framework for understanding how boid snakes in different regions are connected through deep evolutionary history. This framework involved the idea that the separation of major snake lineages occurred by the time period known as the Paleocene at the latest. The Paleocene is a geological epoch that followed the mass extinction event that marked the end of the Cretaceous period. During this epoch, many groups of animals began to diversify and spread into different ecological niches. The interpretation of Titanoboa's position within this timeline placed it in a context that reflects an early stage in the development of Boinae relationships. The placement of Titanoboa within this broader evolutionary framework was examined further in later scientific work published in the year 2015. This study focused on analyzing phylogenetic relationships, which are patterns that show how different species and groups are related through shared ancestry. the analysis used detailed information from fossil structures, especially vertebrae and skull features, to construct a branching pattern of relationships among boid snakes. In this study, Titanoboa was positioned near the base of the Boinae group. The base position in this context refers to an early branching point in the lineage where the species is interpreted as having characteristics that are close to the ancestral form of the group. This placement provided additional information about the early structure of Boinae evolution and the characteristics that may have been present in early members of this group. The phylogenetic analysis, conducted in the year 2015, involved the comparison of many anatomical features across different snake species. These features included bone structure, arrangement of skeletal elements, and details of skull morphology. The data collected from these features were used to construct a branching diagram that represents relationships among species. In this diagram, Titanoboa appeared in a position that reflects its connection to early stages of Boinae development. The study also reinforced the interpretation that Titanoboa holds an important position in understanding how boid snakes diversified over time. The vertebrae of Titanoboa played a central role in these studies. The vertebrae are repeated structures along the spine that provide support and flexibility for movement. In snakes, these bones are especially important because they form the main structure of the body. The shape and articulation of vertebrae can vary among different snake groups, and these variations allow scientists to identify relationships between species. In Titanoboa, the vertebrae show features that match those observed in Boinae snakes. These features include the shape of the neural arch, the arrangement of articulation surfaces, and the proportions of the bone body. Each of these features provides data that contributes to classification and evolutionary interpretation. The skull material that contributed to the classification of Titanoboa includes partial elements that were preserved in fossil form. These elements were examined to identify structural patterns that matched known snake groups. The reduced palatine chonals observed in the skull material are one of the key characteristics used in this classification. These structures are part of the internal arrangement of the skull, and their size and shape vary among different snake groups. In Titanoboa, the reduced condition aligns with patterns seen in Boinae snakes, supporting its placement within that subfamily. The combination of skull and vertebrae evidence provides a more detailed picture of the anatomical features of the animal. The Boinae subfamily itself includes a variety of snake species that share certain anatomical features, particularly in skull structure and vertebral design. These features are used to define the group and distinguish it from other snake subfamilies within Boidae. The distribution of Boinae snakes across the Americas indicates a long history of adaptation to different environments across this region. Fossil evidence shows that members of this group existed in different parts of North and South America, with their range extending across land areas and island regions. The presence of Titanoboa within this group adds to the fossil record that documents the history of Boinae in earlier geological periods. The evolutionary relationships examined in the research of 2013 and 2015 involved detailed comparison of fossil traits with those of living and extinct species. These comparisons focused on identifying shared anatomical features that indicate common ancestry. The analysis included both cranial and postcranial elements, meaning both skull parts and body skeleton parts were studied. Each feature was recorded and evaluated to determine how it relates to other species within Boidae. The resulting phylogenetic structure placed Titanoboa in a position that reflects early branching within Boinae. The geological context in which Titanoboa fossils were found also contributes to understanding its classification. The fossils were recovered from sediment layers that date to a period following the extinction of non-avian dinosaurs. These layers contain remains of many organisms that lived in environments shaped by warm and humid conditions. The preservation of snake vertebrae and skull fragments in these sediments allows for detailed study of anatomical structures. The condition of the fossils provides enough information to identify specific features that are used in classification. The study of Boidae as a family includes examination of many living species that share traits such as elongated bodies, flexible spines and skulls adapted for swallowing prey. Within this family, subfamilies are defined based on differences in skeletal structure and geographic distribution. The Boinae subfamily to which Titanoboa belongs is one of these divisions. The identification of Titanoboa within Boinae is based on a combination of fossil evidence and anatomical interpretation, with vertebrae and skull features providing the main data sources. The research history that includes the year 2013 abstract and the year 2015 study reflects ongoing efforts to understand the relationships among extinct and living snakes. These efforts involve continuous examination of fossil material and refinement of phylogenetic models. The position of Titanoboa within these models remains associated with early Boinae development, supported by structural evidence from fossil remains. The anatomical features observed in Titanoboa continue to be used in studies that explore the early diversification of Boidae snakes across different regions and time periods. The distribution of Boinae snakes across regions such as Mexico, the Antilles and Argentina provides a geographical context for understanding how related species are spread across landmasses. This distribution pattern reflects long-term biological processes that involve movement, adaptation, and separation of populations over extended geological periods. The presence of Titanoboa within this broader framework adds another layer of information to the study of Boinae history, particularly in relation to its placement near the base of the subfamily structure and its connection to early evolutionary stages that were identified through fossil analysis, conducted in the studies from the years 2013 and 2015. The anatomical interpretation of Titanoboa continues to be built from careful examination of fossilized skeletal remains, especially vertebrae and partial skull structures that were preserved in sedimentary rock layers. These remains are studied in detail because snake skeletons are made of many repeating vertebrae, and even small differences in these bones can carry important information about classification. In Titanoboa, the vertebrae show a pattern of structure that includes specific forms of the centrum, which is the main body of each vertebra, along with surrounding processes that connect muscles and support movement. These structures are consistent with traits found in snakes placed within Boinae, supporting the placement of Titanoboa within this subfamily through anatomical comparison. The vertebral morphology includes details such as the thickness of the bone walls, the shape of the articulating surfaces between vertebrae, and the orientation of neural spines. These features are studied by comparing fossil specimens with both modern and extinct snake groups. The comparison process does not rely on external appearance, but instead focuses on internal skeletal traits that remain preserved in fossils. In Titanoboa, these traits show alignment with characteristics used to define Boinae, reinforcing its taxonomic position within the Boidae family. The skull fragments associated with Titanoboa provide additional anatomical information that contributes to its classification. Even though skull fossils are less commonly preserved than vertebrae, the available fragments include key structures that help identify relationships. The reduced palatine chonals observed in the skull material are particularly important. These structures are part of the internal nasal passage system, and their size and shape vary across different snake lineages. In Titanoboa, the reduced form of these openings matches the pattern found in Boinae snakes, which supports the interpretation that it belongs within this group. The combination of skull and vertebral evidence allows researchers to build a more complete anatomical profile of Titanoboa. The skull features provide information about the head structure and internal passages, while the vertebrae provide information about body structure and movement. Together, these two sets of data form the basis for placing Titanoboa within a specific branch of Boidae. This combined evidence strengthens the understanding of its evolutionary position and supports its classification within Boinae as derived from fossil morphology. The broader family Boidae includes a wide range of snakes that share certain structural features, such as elongated bodies, highly flexible spines, and skulls designed for swallowing relatively large prey. Within this family, subfamilies are defined based on differences in skeletal features and geographical distribution. Boinae is one of these subfamilies and is primarily associated with snake species found in the Americas. The members of Boinae show variation in size, habitat, and ecological role, but they share key anatomical traits that allow them to be grouped together in the same subfamily. The distribution of Boinae snakes across regions such as Mexico, the Antilles, and Argentina reflects a long history of dispersal and adaptation across different land environments. Fossil and modern records show that these snakes have existed in both continental and island environments, suggesting that their lineage has been present across a wide range of ecological conditions over long periods of geological time. Titanoboa is placed within this same subfamily based on fossil evidence, which links it anatomically to this group despite its extinction. The scientific interpretation of Titanoboa's relationships has also been influenced by phylogenetic studies that use anatomical data to reconstruct evolutionary connections. In the analysis presented in the year 2013, Titanoboa was examined in relation to other boid snakes from different regions, including taxa associated with Pacific Islands and Madagascar. This analysis focused on identifying shared skeletal features that could indicate common ancestry or deep evolutionary relationships among geographically separated groups. The results suggested that Titanoboa shared certain structural characteristics with these groups, providing information about how boid snakes may be related across different regions of the world. This interpretation introduced the idea that major divisions within Boidae may have separated earlier than previously assumed, with divergence events occurring by the Paleocene epoch at the latest. The Paleocene epoch followed the major extinction event that ended the Cretaceous period and marked a time when many animal groups began to diversify and expand into new ecological roles. In this context, Titanoboa was placed near an early branching point within Boinae, suggesting that it represents a form close to the ancestral structure of the group based on anatomical traits preserved in fossils. Further analysis conducted in the year 2015 refined the understanding of Titanoboa's placement within Boinae. This study used a broader set of anatomical data and a more detailed phylogenetic framework to examine relationships among boid snakes. The analysis included comparisons of vertebral structure, skull morphology, and other skeletal features across multiple species. The resulting phylogenetic model placed Titanoboa near the base of Boinae, indicating that it represents one of the earlier diverging lineages within this subfamily. The basal position assigned to Titanoboa in this phylogenetic structure is based on the presence of certain anatomical features that are interpreted as closer to ancestral conditions within Boinae. These features are identified through systematic comparison of fossil traits across multiple species. The study's conclusions are supported by consistent patterns observed in vertebral and cranial anatomy, which align Titanoboa with early stages in the evolutionary development of Boinae snakes. The vertebral column of Titanoboa is especially important in understanding its classification, because snake evolution is often studied through spinal structure. Each vertebra contains multiple anatomical regions, including the centrum, neural arch, and various processes that connect muscles and joints. The arrangement and proportions of these structures differ between snake groups, allowing researchers to identify evolutionary relationships. In Titanoboa, the vertebral characteristics align closely with those seen in Boinae snakes, supporting its placement within this subfamily. The skull evidence, although fragmentary, contributes key information about cranial anatomy. The reduced palatine chonals observed in Titanoboa are part of a pattern seen in Boinae snakes, where certain internal skull openings are smaller or differently shaped compared to other snake groups. This trait is used as one of several anatomical indicators in classification. When combined with vertebral data, it strengthens the interpretation of Titanoboa's taxonomic position. Titanoboa possessed extraordinary hunting abilities and tremendous physical strength. Its success as a predator was not based on speed or venom. Instead, it relied on its immense size, powerful muscles, patience, stealth, and the ability to overpower prey through sheer force. Every part of its body was suited for capturing and killing animals in the warm tropical ecosystems where it lived. The enormous size of Titanoboa played a central role in its hunting methods. Fossil evidence suggests that this snake could reach lengths of around 42 feet and weigh more than 1 ton. Such dimensions made it one of the largest snakes ever known to science. Its massive body contained powerful muscles extending from its head to the tip of its tail. These muscles provided the force necessary to move its great weight through water and across land, while also allowing it to capture struggling prey. Scientists believe that Titanoboa spent much of its life near rivers, swamps, and wetlands. Water offered several advantages for a snake of such immense size. On land, moving a body weighing more than a ton would require considerable energy. In water, however, buoyancy helped support its weight, making movement easier and more efficient. Rivers and swamps also attracted many animals searching for water, food, and shelter. This created ideal hunting grounds for a large ambush predator. Ambush hunting was likely one of Titanoboa's primary techniques. Rather than chasing prey over long distances, the snake probably relied on remaining hidden and waiting for an opportunity to strike. This strategy is common among many large snakes living today, and would have been especially effective for a predator as massive as Titanoboa. The dense vegetation of Paleocene forests provided excellent cover. Thick plants, fallen logs, tangled roots, and muddy riverbanks created countless places where a giant snake could conceal itself. Titanoboa may have remained almost motionless for long periods while waiting for prey to approach. Patience would have been one of its greatest hunting tools. Many animals are less likely to notice danger when it remains still. A giant predator hidden among vegetation could become surprisingly difficult to detect. Titanoboa's coloration remains unknown because skin pigments are rarely preserved in fossils. However, Many scientists suspect that its appearance may have helped it blend into its surroundings. Dark, earthy colors would have made it even harder for prey to spot the snake among shadows, mud, and plant cover. When prey moved within range, Titanoboa would have launched a rapid strike. Despite its enormous size, the front part of its body was likely capable of moving with remarkable speed. Modern large snakes demonstrate that significant body mass does not prevent quick striking movements. The neck and head can be propelled forward rapidly using powerful muscles stored along the body. The initial strike was one of the most important moments in the hunt. A successful strike allowed Titanoboa to seize prey before it could escape. Its jaws contain numerous sharp, curved teeth designed for gripping rather than cutting. These teeth pointed backward, making it difficult for captured animals to pull free. Every struggle would push the prey deeper into the snake's grasp. Unlike venomous snakes, Titanoboa probably did not inject toxins into its victims. There is no evidence suggesting that it possessed venom glands capable of killing large prey. Instead, its hunting success came from physical strength. Once the prey was secured in its jaws, the snake would have used the rest of its body to overpower the victim. Constriction was likely the primary killing method employed by Titanoboa. This technique involves wrapping the body around prey and applying intense pressure. Modern boas and pythons use this method very effectively, and Titanoboa is believed to have done the same on a much larger scale. The process likely began immediately after the strike. As soon as the prey was grabbed, Titanoboa would have thrown loops of its muscular body around the animal. The snake's great length allowed it to form multiple coils around large victims. Once these coils were in place, the pressure would begin increasing. For many years, people believed constrictor snakes killed by crushing bones. Modern research has shown that this is usually not the case. Instead, constriction rapidly interferes with blood circulation and breathing. Pressure from the snake's coils prevents blood from moving normally through the body. Vital organs receive less oxygen, and unconsciousness can occur surprisingly quickly. A giant constrictor such as Titanoboa would have generated extraordinary force. Its immense muscle mass suggests that the pressure exerted by its coils could have been far greater than that produced by living snakes. Large prey trapped within those coils would have found escape extremely difficult. Every movement made by the prey would have worked against it. When an animal exhaled, the coils would tighten slightly. This prevented the chest from expanding fully during the next breath. As the struggle continued, breathing became increasingly difficult. Blood circulation also became impaired, causing weakness and loss of strength. Titanoboa's muscular power was likely among its most deadly features. The snake's body was essentially a long chain of interconnected muscles, capable of working together. These muscles not only provided movement, but also generated the force necessary for constriction. The larger the body, the greater the potential strength. Scientists studying fossil vertebrae have discovered that Titanoboa possessed exceptionally robust skeletal structures. Its vertebrae supported powerful muscles and allowed the body to withstand significant stress during hunting. Every coil wrapped around prey represented a coordinated effort involving large portions of the snake's body. The snake's strength would have allowed it to tackle a wide range of animals. Fossils found in the same geological formations indicate the presence of fish, turtles, crocodile relatives, and numerous other creatures. Some of these animals may have become prey for Titanoboa. Fish likely formed an important part of its diet. Rivers and wetlands would have contained abundant aquatic life, providing a reliable food source. Capturing fish required a somewhat different approach than hunting land animals. In water, Titanoboa may have relied on stealth and sudden lunges. The snake could remain hidden beneath the surface, with only small portions of its body exposed. Water helped conceal movement. A fish swimming nearby might not notice the predator until it was too late. A rapid strike from a giant snake could capture large aquatic prey before it had a chance to escape. Turtles may also have been among Titanoboa's prey. Fossils from the same period reveal the existence of sizable turtle species inhabiting these ancient wetlands. While a turtle's shell provided protection against many predators, Titanoboa's enormous strength may have enabled it to overcome such defenses. The snake could have seized vulnerable body parts extending beyond the shell and used constriction to immobilize the animal. Crocodile relatives represented another potential food source. The Paleocene wetlands were home to several crocodilian species. These reptiles were themselves dangerous predators, yet a fully grown Titanoboa may have possessed enough size and power to challenge them. Ambush attacks from concealed positions near water could have given the snake an advantage. The relationship between Titanoboa and crocodilian species was likely complex. Sometimes they may have competed for similar prey. At other times, larger individuals of Titanoboa may have hunted smaller crocodilians. Likewise, young Titanoboa may have faced risks from crocodilian predators before reaching full size. The ability to remain hidden was crucial for successful hunting. Titanoboa probably spent much of its time conserving energy. Large predators often benefit from avoiding unnecessary activity. Moving enormous distances requires substantial energy, especially for animals with massive bodies. By waiting patiently near areas frequented by prey, the snake increased its chances of success while minimizing effort. Its sensory abilities likely played a major role during hunts. Modern snakes gather information about their surroundings through several specialized systems. Although soft tissues are not preserved in fossils, scientists believe Titanoboa possessed sensory organs similar to those found in living relatives. The tongue was probably an important tool. Snakes repeatedly flick their tongues to collect tiny particles from the air and ground. These particles are transferred to a sensory structure in the mouth known as the Jacobson's organ. This system helps snakes detect chemical traces left behind by other animals. Titanoboa likely used chemical signals to locate prey and identify areas where animals frequently traveled. Even in dense forests where visibility was limited, scent information could reveal the presence of potential meals. Vibrations may also have helped the snake detect nearby movement. Snakes are sensitive to vibrations traveling through the ground and water. Footsteps, splashing, and other disturbances create signals that can be detected by a waiting predator. A large animal approaching a riverbank would produce vibrations that Titanoboa might recognize long before seeing the prey directly. Vision probably contributed to hunting as well. While scientists cannot determine exactly how good Titanoboa's eyesight was, many snakes possess adequate vision for detecting movement. Combined with chemical sensing and vibration detection, this would have created an effective system for locating prey. Water-based ambushes may have been especially important. Many modern large constrictors spend considerable time in aquatic environments. Water allows them to approach prey quietly and reduces the effort required to support their body weight. Titanoboa's immense size suggests that rivers and swamps were likely central to its hunting lifestyle. A hidden position beneath the water's surface would have allowed the snake to approach animals drinking at riverbanks. Mammals, birds, reptiles, and other creatures must eventually come to water. This predictable behavior creates opportunities for ambush predators. The attack itself may have occurred within seconds. After detecting suitable prey, Titanoboa could remain motionless until the target moved close enough. Then the snake would surge forward, seize the victim, and immediately begin wrapping its body around it. The suddenness of the attack reduced the prey's chances of escape. Large prey often struggle violently when captured. Titanoboa's powerful muscles would have been essential during these moments. The snake needed to maintain control despite kicks, bites, claws, or other defensive actions. Multiple coils distributed pressure across the victim's body while keeping it securely restrained. Once the prey stopped moving, swallowing could begin. Snakes possess highly flexible jaws that allow them to consume animals much larger than their heads. Titanoboa likely shared this ability. Special adaptations in the skull and jaw joints permitted gradual swallowing over extended periods. The process required patience. The snake would position the prey head first whenever possible, reducing resistance from limbs, scales, or other body structures. Powerful throat muscles then helped pull the meal deeper into the digestive system. The enormous size of Titanoboa meant that it could consume substantial meals. A single successful hunt may have provided enough food to sustain the snake for long periods. Modern large snakes can survive for weeks or even months between meals, depending on prey size and environmental conditions. Titanoboa may have exhibited similar patterns. Digestion itself required considerable energy. Warm environmental temperatures likely aided this process. Reptiles depend heavily on external heat sources, and the tropical climate of Paleocene South America would have provided favorable conditions for processing large meals. Young Titanoboas probably employed hunting methods similar to those of adults, although on a smaller scale. Juveniles would have targeted smaller prey appropriate to their size. As they grew larger and stronger, they could gradually expand the range of animals they hunted. Growth offered important advantages. A larger body increased strength, expanded hunting opportunities, and reduced vulnerability to predators. Reaching adulthood transformed Titanoboa into one of the dominant predators within its ecosystem. The snake's deadly strength was not limited to constriction alone. Moving such a massive body required exceptional muscular development. These same muscles allowed the animal to navigate rivers, climb over obstacles, and maintain control during encounters with prey. Every aspect of its lifestyle depended on physical power. The structure of its ribs also contributed to hunting success. Snakes possess numerous ribs extending along much of their bodies. These ribs support muscles involved in movement and constriction. Titanoboa's large frame meant that this system operated on an enormous scale, creating a highly effective mechanism for generating force. Researchers studying living constrictors have observed how muscle contractions occur in coordinated patterns. Rather than squeezing randomly, snakes carefully adjust pressure in response to prey movements. Titanoboa likely possessed similar control. Such coordination allowed maximum effectiveness while conserving energy. A struggling animal might attempt to twist, kick, bite, or pull away. The snake's nervous system and muscular control enabled it to respond instantly, maintaining secure coils around the victim. This adaptability increased hunting success and reduced the risk of injury. Injury avoidance was important for any large predator. A damaged jaw, broken tooth, or serious wound could threaten survival. Ambush hunting reduced many risks because prey often had little opportunity to fight back before becoming restrained. Titanoboa's environment likely influenced its hunting strategies in many ways. Seasonal flooding may have altered prey distribution. Rising waters could concentrate animals in certain locations or create new aquatic habitats rich in fish and other prey species. The snake's flexibility allowed it to exploit changing conditions. The warm climate may also have contributed directly to its extraordinary size and strength. Reptiles depend on environmental temperatures to regulate many bodily functions. Consistently warm conditions support growth, activity, and digestion. These factors may have helped Titanoboa reach dimensions rarely seen among snakes. Fossil discoveries indicate that Titanoboa existed within ecosystems filled with abundant food resources. Rich wetlands supported diverse animal communities, providing numerous hunting opportunities. Such environments were ideal for sustaining a predator with substantial nutritional requirements. Scientists continue investigating exactly how Titanoboa lived and hunted. Each new fossil provides additional clues about its behavior, anatomy, and ecological role. Although many details remain uncertain, evidence strongly suggests that Titanoboa combined stealth, patience, sensory awareness, constriction, and overwhelming muscular strength to capture prey. Its hunting success depended on a carefully integrated set of abilities. Concealment allowed it to approach prey unnoticed. Sensitive detection systems helped locate potential victims. Rapid strikes secured prey before escape. Powerful coils immobilized and killed captured animals. Flexible jaws enabled the consumption of large meals. Strong muscles supported every stage of the hunting process. The deadly strength of Titanoboa originated from its immense size and specialized anatomy. Powerful vertebrae, extensive muscle networks, numerous ribs, and a body adapted for both land and water combined to create one of the most formidable predators of the Paleocene world. Hidden within the tropical wetlands of ancient South America, this giant snake relied on patience, precision, and overwhelming physical force as it hunted among rivers, swamps, and forests where countless animals moved through an environment shaped by warmth, abundant vegetation, and thriving aquatic ecosystems. The diet of Titanoboa was based on animals that lived in water and around water. The environment where it lived had many slow-moving rivers, swamp areas, and shallow lakes. These water systems were home to a wide variety of animals. Fish were present in large numbers and came in different forms and sizes. Many fish lived near the surface of the water, while others stayed closer to the bottom. Titanoboa had access to these fish because of its strong connection to water environments. Its body structure suggests that it spent a large amount of time in water or near water, which allowed it to approach aquatic animals with ease. Fish formed a part of its food intake. The way fish move in water, the way they gather in groups, and the way they rely on water currents made them available to a snake that could remain still and wait for the right moment. Titanoboa likely used slow movements and patience to position itself in areas where fish were active. When a fish came close enough, it could be taken quickly. The swallowing process for fish would have been easier compared to other animals because of their shape and flexibility. Fish also provided a steady source of food in environments where water was present throughout the year. Another important part of Titanoboa's diet came from reptiles that lived in the same region. One group of reptiles that played a major role in its diet were crocodilian animals. These animals lived in rivers and swamps and spent much of their time in water. They had strong bodies, thick skin, and powerful jaws. Young crocodilian animals were especially important in the feeding habits of Titanoboa. These younger reptiles were smaller in size and often spent time near riverbanks or in shallow waters where they were easier to approach. Titanoboa likely used stealth in water to get close to them before striking. The structure of its jaws allowed it to hold on to prey, and its long body helped it wrap around animals to control their movement. Adult crocodilian animals were also part of the environment, and although they had strong defense abilities, Titanoboa had access to them at certain times. The feeding on these animals likely depended on opportunity, water conditions, and the situation of the encounter. In periods when crocodilian animals were resting or not fully alert, Titanoboa could take advantage of the situation. The swallowing process for reptiles required time and energy, and Titanoboa had a body designed to handle large meals that could sustain it for long periods. Turtles were also present in the same environment and formed another part of the diet. These turtles lived in water and often moved slowly on land or in shallow areas. Their shells provided protection, but their movement patterns made them predictable in certain conditions. Titanoboa could encounter turtles in water or near the edges of water bodies. When dealing with turtles, the snake would need to use methods that allowed it to access the soft parts of the body. This involved positioning and control of the animal. Turtles provided a source of nutrients that contributed to the energy needs of Titanoboa. The feeding behavior of Titanoboa also involved a wide range of other animals that lived in swamp and river environments. Amphibians were common in these regions. These included large frogs and similar animals that lived both in water and on land. Amphibians have moist skin and often gather in water during different stages of their life cycle. Titanoboa could encounter them while they were in water or near muddy areas. These animals were part of its food intake because of their presence in large numbers and their accessibility. Small reptiles that lived in the same environment also contributed to its diet. These reptiles moved through vegetation, water edges, and muddy ground. Titanoboa's ability to move through water and shallow areas allowed it to come into contact with these animals. When it encountered them, it could use quick movement to secure them. The structure of its mouth allowed it to take in animals in one piece, which is a characteristic of large snakes that feed on whole prey. Birds that lived near water areas were also part of the ecosystem. These birds often rested on low branches, river edges, and swamp vegetation. Some birds fed on fish and amphibians, which brought them close to water surfaces where Titanoboa was present. When birds came near the water, they became part of the feeding opportunities available to the snake. The timing of feeding would depend on movement patterns of birds and their behavior near water. Titanoboa's feeding process involved the ability to take in large amounts of food at one time. After capturing prey, it would use its jaws to hold the animal and then begin the process of swallowing. The swallowing process required coordination of muscles along its body. The prey would move through the body slowly. This process allowed Titanoboa to consume animals that were large in size relative to many other snakes that exist in modern times. After feeding, the snake would enter a period of rest while digestion took place. This resting period could last for long durations depending on the size of the meal. The structure of Titanoboa's skull and teeth played a role in its diet. Its teeth were curved and designed to hold prey firmly. They were not designed for chewing because snakes do not chew their food. Instead, the teeth helped keep the prey in place while it was being swallowed. The jaws were flexible, allowing them to open wide enough to take in animals whole. This feature allowed Titanoboa to feed on a wide range of animals found in its environment. Water played an important role in feeding behavior. Titanoboa likely spent a large portion of its time in water, where it could move more easily and remain hidden. Water also helped support its body weight and allowed it to move without creating strong disturbance on the surface. This made it easier to approach prey without being noticed. Many of the animals it fed on were also dependent on water, which increased the chances of encounters. The availability of food in its environment was influenced by seasonal changes in rainfall and water levels. During the periods of high water, rivers and swamps expanded, spreading animals across wider areas. This created more space for feeding opportunities. During periods of lower water levels, animals became more concentrated in smaller areas, which also affected feeding behavior. Titanoboa adapted to these changes by following water systems and remaining close to environments where prey was present. The digestive system of Titanoboa was capable of handling large meals. After consuming prey, the food would be broken down slowly inside the body. The energy gained from these meals supported movement, growth, and survival. Because feeding events could provide large amounts of energy at one time, there would often be long intervals between meals. During these intervals, Titanoboa would remain in areas where it could conserve energy while waiting for another opportunity to feed. The interaction between Titanoboa and its prey was based on timing and positioning. Many of the animals it fed on had behaviors that brought them close to water surfaces or shallow areas. These behaviors created opportunities for encounters. Titanoboa relied on staying still for long periods and moving only when necessary. This allowed it to reduce detection by animals in the environment. The structure of its body allowed it to move through both deep water and shallow swamp areas. This flexibility gave it access to different types of prey across different parts of its habitat. It could remain partially hidden in muddy water or submerged vegetation while waiting for movement nearby. The feeding method involved sudden action, followed by strong control of the prey using its body. The relationship between Titanoboa and its environment was closely linked to the availability of aquatic and semi-aquatic animals. Rivers, swamps, and floodplains provided continuous sources of food. Fish populations, crocodilian populations, turtle populations, amphibian populations, and small reptiles formed a connected system of available food sources. Titanoboa moved through this system and used it for survival through feeding. The fossil evidence that supports understanding of its diet comes from the region where remains of Titanoboa were found alongside remains of other animals. These fossils include bones of reptiles and other creatures that lived at the same time. The structure of the environment shows that water played a central role in shaping the food web. Titanoboa was part of this food web, using its abilities to feed on animals that lived in or near water. The skull shape and jaw flexibility give further evidence of its feeding habits. The wide opening of the mouth allowed it to take in prey without breaking it into smaller pieces. The muscles involved in swallowing were strong and worked in coordination with the rest of the body. This made it possible for Titanoboa to consume animals that required extended swallowing time. The role of temperature in the environment also influenced feeding behavior. Warm conditions supported the activity of reptiles and other animals that formed part of its diet. High levels of warmth allowed reptiles to remain active throughout much of the time, which increased the chances of encounters between predator and prey. The constant activity of aquatic environments provided steady feeding opportunities. The feeding patterns of Titanoboa were also influenced by its need for energy conservation. After consuming a meal, it would remain inactive for long periods while digestion continued. This reduced the need for frequent hunting. The cycle of feeding and resting formed a pattern that repeated over time based on availability of prey. The presence of dense vegetation in swamp areas provided hiding places for both prey and Titanoboa. This vegetation reduced visibility in water and on land edges. Animals moving through these areas had limited visibility, which created conditions where encounters could occur unexpectedly. Titanoboa used these conditions to position itself for feeding. The life of Titanoboa in its environment was centered around water systems, and its diet reflected the animals that lived in those systems. Fish, Crocodilian animals, turtles, amphibians, small reptiles and water-associated birds formed the main sources of food. Each of these groups contributed to the overall feeding pattern of Titanoboa across time, with availability changing based on location and environmental conditions. The world that allowed giant reptiles to live and spread was shaped by long periods of stable warmth, high humidity, and strong natural cycles of water and plant growth. These conditions were not short events, but lasted for very long spans of Earth history. The land, rivers, and shallow seas all took part in building an environment that supported large cold-blooded animals. The air, water, soil, and living plants worked together in a connected system that kept heat and moisture present in many regions at the same time. The atmosphere held a high level of warmth during these ancient times. Heat was present across many regions of the planet throughout the year. Seasonal changes in temperature were weak in many places, so the environment did not shift strongly between cold and hot periods. This steady warmth allowed reptiles, which depend on outside heat sources to regulate body functions, to remain active for long periods of time without facing strong cold stress. The constant presence of heat also supported long growing periods for plants, which formed the base of food systems that supported many living creatures. Moisture in the air was also present in large amounts. Rainfall occurred frequently across many land areas, feeding rivers, wetlands, and forested regions. Water cycles moved steadily between oceans, land, and atmosphere. Evaporation from warm waters added more moisture into the air, and this moisture returned as rain over land surfaces. This repeating cycle kept many regions wet for long stretches of time. Wet ground conditions supported dense plant growth, which provided shelter and feeding areas for many animals that lived in these environments. The oceans played an important role in maintaining stable heat conditions. Large ocean surfaces stored warmth and released it slowly into the surrounding air. Ocean currents moved this heat across different regions of the planet, spreading warm conditions into areas that might otherwise have been less stable in temperature. Shallow seas covered parts of land in many regions, creating warm water zones rich in life. These marine environments also supported food chains that connected aquatic and land systems. Carbon levels in the atmosphere were higher during these ancient periods. This influenced the amount of heat held within the atmosphere. Higher carbon levels supported the trapping of solar energy within the air layer surrounding the planet. This contributed to steady warmth across both land and sea surfaces. Plant life responded strongly to these conditions, expanding across many regions. Dense plant cover developed in many habitats, ranging from low wet areas to forested zones. Vegetation growth was continuous in many regions due to the long warm seasons. Plants did not experience long pauses in growth, and this allowed ecosystems to build thick layers of plant material over time. Leaves, stems, and roots formed complex ground systems that supported insects, small animals, and large reptiles. Fallen plant material added nutrients to soil, which supported new plant growth. This cycle continued in a steady pattern, strengthening the natural base that supported larger animals. River systems were wide and slow-moving in many areas. Constant rainfall and steady heat fed these rivers, keeping water levels high for long periods. Floodplains formed along river banks, creating flat areas with rich soil. These floodplains supported dense vegetation and provided water access for land animals. Wetlands formed in many low areas, holding water for long durations. These wetlands became important zones for feeding, resting, and movement of large reptiles. The land surface in many regions remained soft and rich in organic material due to continuous plant decay and water flow. Soil layers were often deep and held large amounts of moisture. This supported root systems of plants that grew steadily without interruption. In many places, ground conditions allowed both aquatic and land-based environments to exist close to each other. This close arrangement of habitats made it easier for large reptiles to move between water and land areas in search of food and suitable living conditions. Air circulation patterns distributed heat and moisture across large distances. Warm air moved across continents and oceans, carrying humidity into different regions. This movement supported rainfall patterns that remained active across wide areas. Cloud formation occurred frequently due to the presence of moisture and heat in the atmosphere. Clouds contributed to regular rainfall events, which maintained wet conditions on land surfaces. Sunlight reached the surface in strong and consistent amounts. Plant life responded to this energy through continuous growth processes. Photosynthesis occurred at high levels due to the combination of sunlight, water, and carbon availability. This supported the expansion of vegetation across many landscapes. Plant density increased in regions with stable water access, forming environments rich in cover and food resources. Wet forests formed in many regions where water supply remained constant. These forests had tall plant structures, dense lower growth, and layered vegetation. The ground in these forests often remained moist throughout long periods. Fallen plant matter decomposed quickly due to warm and wet conditions, returning nutrients to the soil and supporting further plant growth. These environments supported many types of reptiles, including large species that required stable conditions for movement and feeding. Lowland regions near rivers and seas developed into productive ecosystems. These areas had constant access to water and nutrients carried by flowing rivers. Sediment deposits from water movement created fertile ground. Plants grew thickly in these regions, and animal life concentrated around available water sources. Large reptiles used these areas for feeding and movement due to the availability of prey and stable environmental conditions. Wetland systems spread across wide regions of land. These wetlands held water for long periods and supported both aquatic and semi-aquatic life forms. Plant growth in wetlands was dense and adapted to constant moisture. Soft ground conditions allowed large reptiles to move through these areas with ease. Food sources were abundant in wetlands due to the presence of fish, amphibians, and smaller reptiles. Shallow water environments extended over parts of land where sea levels reached inland areas. These shallow waters were warm and supported rich biological activity. Marine plants and small organisms formed the base of aquatic food systems. Larger reptiles used these environments for hunting and feeding, as well as for movement between different zones of water and land. The balance of oxygen in the atmosphere supported active biological systems. Air composition allowed animals and plants to maintain steady metabolic processes. Reptiles, which rely on external heat sources, benefited from the consistent environmental conditions that allowed their body systems to function without interruption caused by cold stress. This supported longer periods of movement and feeding activity. Storm systems developed in regions where heat and moisture interacted strongly. These storms brought heavy rainfall to land surfaces. Rainfall contributed to river flow, soil moisture, and plant growth. Storm patterns were part of the natural water cycle that kept ecosystems active. After storms, water spread across land surfaces, replenishing wetlands and river systems. Long periods of stable climate conditions allowed ecosystems to develop without strong interruptions. Plant systems expanded continuously, and animal populations adjusted to steady environmental conditions. Reptiles that required external heat sources found suitable conditions across many regions, allowing them to spread widely. The availability of food resources remained steady due to continuous plant growth and aquatic life development. Soil chemistry in many regions remained rich in organic content. Decomposed plant material added nutrients to the ground. Microorganisms in the soil broke down organic matter and released nutrients that supported new plant growth. This cycle continued without interruption in many warm and wet regions, maintaining strong plant productivity over long periods. Freshwater systems remained active across many land areas. Lakes formed in low-lying regions and collected water from rainfall and river flow. These lakes supported aquatic ecosystems and provided drinking sources for land animals. Water levels in lakes often changed slowly, allowing stable environments to form around them. Coastal regions were shaped by the interaction between land and sea. Tides and wave activity influenced shoreline conditions. Nutrient exchange between land and sea supported diverse biological systems. Coastal wetlands developed in many areas, providing habitats for a wide range of species. Heat distribution across the planet was influenced by solar energy reaching the surface and being absorbed by land and water. Water bodies stored heat and released it gradually. This created stable thermal conditions in many regions. Land areas near water bodies benefited from this stability, supporting continuous biological activity. Atmospheric moisture levels supported cloud formation on a regular basis. Clouds influenced rainfall patterns and helped maintain humidity in many ecosystems. Humid conditions supported plant growth and reduced the risk of dry periods in many regions. This allowed vegetation to remain active for long durations. Plant diversity increased in regions where water, heat, and soil nutrients were abundant. Different plant types occupied various layers of ecosystems. Some plants grew in water-rich soil, while others occupied higher ground. This variety of plant life supported a wide range of animal species that depended on vegetation for shelter and food. Large reptiles occupied many ecological zones, including rivers, wetlands, forests, and coastal areas. Their presence in these environments was supported by the availability of warm conditions and steady food sources. Movement between water and land environments was common in many species, supported by the structure of these ecosystems. Energy flow within ecosystems remained steady due to continuous plant growth and animal activity. Sunlight provided energy to plants, which supported herbivorous animals, which in turn supported carnivorous species. This flow of energy remained active throughout long periods due to stable environmental conditions. Water availability across land areas remained high due to frequent rainfall and river systems. This allowed animals to access water sources without long interruptions. Wet environments supported reproduction and feeding activities for many species of reptiles. The interaction between air, water, land, and living organisms created stable environmental conditions over long durations. Each component supported the others through continuous cycles of exchange. Heat and moisture remained present across many regions, supporting the growth of plants and the survival of large reptiles that depended on these conditions for daily activity and long-term existence. The environment where Titanoboa lived had steady high temperatures that supported reptile life and influenced how body processes worked. Reptiles depend on external heat to regulate body temperature, so the climate conditions played an important role in daily activity patterns. The warm water bodies and surrounding land areas created spaces where Titanoboa could move through rivers, swamps, and forested wetlands. These habitats also supported fish populations, amphibians, turtles and other reptiles that formed the feeding sources within the ecosystem. Titanoboa occupied aquatic and semi-aquatic zones for most of its life cycle. Its movement through water allowed it to travel across different parts of river systems without much resistance from vegetation. In these water systems, it could remain partially hidden while observing movement of animals in the environment. The structure of its body supported slow but powerful movement, allowing it to remain still for long periods and then shift position when needed for feeding or relocation. The feeding activity of Titanoboa was closely connected to the availability of aquatic organisms. Fish populations formed a steady food supply, and large fish species provided substantial energy intake. Amphibians living near water edges also formed part of its feeding pattern. Turtles in shallow water zones were part of the available animal life in the ecosystem, and Titanoboa had the ability to consume them based on jaw flexibility and body structure that supported expansion during feeding. The ecosystem surrounding Titanoboa included a wide range of plant life that supported food chains through decomposition and nutrient recycling. Fallen leaves, decaying plant material, and organic matter created nutrient-rich sediments in riverbeds and swamp areas. These sediments supported algae and microorganisms that formed the base of aquatic food networks. Small aquatic animals fed on these organisms, and larger aquatic animals fed on smaller organisms, creating multiple layers of energy transfer that Titanoboa accessed at upper levels of the feeding structure. Titanoboa's presence in river systems influenced movement patterns of other animals. Many species of reptiles and amphibians adjusted their behavior around water areas where Titanoboa was active. The timing of movement into shallow water, riverbanks, and flooded zones was affected by awareness of predator presence. Some animals used dense vegetation along water edges for cover, while others remained in deeper water areas, depending on feeding needs and safety behavior. The physical structure of Titanoboa included strong muscle development along its entire body length. This muscle arrangement allowed it to move through water and land edges with controlled force. The body contained a large number of vertebrae that supported flexibility and strength during movement. Its skull structure supported wide jaw opening capability, which allowed it to take in prey of substantial size relative to its own head structure. Teeth were curved inward to maintain grip during feeding. The digestive system of Titanoboa was adapted to process large food intake events. After feeding, it could remain inactive for extended periods while digestion took place. This reduced energy usage during times when food was already consumed. The metabolic rate was influenced by environmental temperature, meaning that warm conditions supported more active digestion processes. The ecosystem temperature allowed this biological function to operate at consistent levels throughout the year. The river systems where Titanoboa lived had seasonal changes in water levels. During periods of high water, floodplains expanded and created temporary aquatic zones. These expanded zones increased access to food sources and allowed Titanoboa to move into new areas. During lower water levels, movement became concentrated within deeper channels and permanent water bodies. These cycles affected distribution of aquatic animals and influenced feeding patterns. The plant life in surrounding regions included large trees, swamp vegetation, and fast-growing plants adapted to warm and wet conditions. These plants contributed to shelter formation along riverbanks. Root systems helped stabilize soil along water edges, reducing erosion and creating structured environments for small and medium-sized animals. Fallen trees created barriers and resting zones within water channels. which influenced movement routes for aquatic creatures. Titanoboa interacted with other large reptiles that lived in the same period. Crocodile-like reptiles inhabited rivers and swamp zones, and turtles with heavy shells were present in both shallow and deeper waters. These animals shared similar habitats, but occupied different positions within feeding networks. Titanoboa accessed similar environments, but used different movement patterns and feeding strategies based on its body structure and hunting method. The hunting process of Titanoboa relied on ambush behavior. It could remain still in water or near water edges for long periods while waiting for movement from nearby animals. When an animal came within reach, it could use rapid body motion to secure contact. Once contact was made, the body could wrap around the target and apply pressure through muscle contraction. This process reduced movement from the captured animal until feeding could begin. The ecosystem energy flow included constant recycling of organic material. Dead organisms decomposed in water and land areas, returning nutrients to soil and sediment. Microorganisms played a role in breaking down organic matter, which supported plant growth. This plant growth supported herbivorous animals, which then supported carnivorous animals at higher feeding levels. Titanoboa existed within this structure as a major aquatic predator, influencing distribution of prey species. Fossil evidence of Titanoboa was discovered in rock formations located in what is now Colombia. These fossils included vertebrae and rib fragments that allowed scientists to estimate body structure and movement ability. The fossil layers also contained remains of plants and other animals, giving information about the environment at the time. Sediment composition suggested a warm and humid climate with frequent water coverage. The water system supported continuous movement of nutrients and organisms. Rivers carried sediments across large areas, depositing materials in floodplains and low-lying regions. These deposits created fertile zones that supported plant growth and animal life. The constant flow of water also supported migration routes for aquatic animals, which created feeding opportunities for predators such as Titanoboa. Titanoboa's activity pattern was influenced by environmental stability. Warm conditions allowed consistent movement without seasonal cold periods that would reduce activity levels. This stability supported continuous feeding cycles and interaction with aquatic life throughout the year. The availability of prey organisms remained relatively steady due to continuous reproduction cycles in warm water environments. The sensory system of Titanoboa included detection of movement and vibration in water. This ability allowed it to sense nearby animals even when visibility was limited by water conditions or vegetation cover. Chemical sensing through tongue movement provided additional environmental information, helping it locate prey and navigate water systems. These sensory inputs supported accurate positioning within its habitat. Growth of Titanoboa occurred over long periods, with body development influenced by food availability and environmental conditions. Juvenile stages involved smaller body size and feeding on smaller aquatic organisms. As growth continued, feeding capability expanded, allowing access to larger prey types within the ecosystem. Growth rate was connected to temperature and resource availability in the environment. The interaction between Titanoboa and other organisms contributed to regulation of population distribution within aquatic systems. Prey populations adjusted reproduction and movement patterns in response to predation pressure. This created balance in aquatic food networks where energy transfer remained active through multiple feeding levels. Vegetation and microbial life supported this balance through continuous nutrient cycling. River edges and swamp regions formed transitional zones between land and water environments. These zones supported mixed communities of animals that depended on both land and aquatic resources. Titanoboa used these transitional areas for movement and feeding activity. Dense plant growth in these zones provided cover and supported ambush behavior during feeding events. The structure of the ecosystem included repeated cycles of flooding and water retreat. These cycles reshaped land surfaces, redistributed nutrients, and changed habitat boundaries. Titanoboa adapted to these changes by moving across different water channels and floodplain regions. This flexibility in movement allowed access to varied feeding areas within the same geographic region. Aquatic plant life contributed oxygen and organic material to water systems. Floating vegetation and submerged plants provided habitat for small aquatic organisms. These organisms formed part of the feeding network that supported larger animals. Titanoboa operated within this system by feeding on organisms located at different levels of aquatic life. The presence of large reptiles in river systems created zones of activity where movement patterns overlapped. These zones included areas of dense vegetation, open water channels, and shallow floodplains. Titanoboa moved through these zones based on feeding needs and environmental conditions. The distribution of animal life across these zones shaped the structure of ecological interactions. Sediment layers in fossil sites show repeated cycles of deposition from water flow. These layers contain organic material that indicates presence of plant and animal life over long periods. The preservation of bones within these layers provides information about body structure and environmental conditions. The arrangement of fossils suggests that aquatic systems played a central role in ecosystem structure. The life cycle of Titanoboa was closely linked to water availability. Water systems provided movement routes, feeding opportunities, and temperature regulation. The ecosystem supported continuous interaction between predator and prey organisms through stable environmental conditions. This stability allowed long-term development of aquatic food networks. Organic matter breakdown in water and soil supported microbial growth, which formed the base of nutrient cycles. These cycles supported plant growth along riverbanks and floodplains. Plant growth supported herbivorous organisms, which contributed to energy transfer through aquatic and terrestrial systems. Titanoboa accessed energy within this network through feeding on aquatic animals. Movement of Titanoboa through water involved coordinated muscle activity along its body. This movement allowed navigation through complex environments with vegetation, debris, and water currents. Its body structure supported both slow positioning and rapid movement when required for feeding interaction. This combination of movement types supported survival within aquatic habitats. Environmental conditions remained consistently warm, supporting reptile physiology and activity levels. This allowed continuous participation in ecosystem processes, without interruption, from temperature changes that reduce reptile activity. Aquatic and terrestrial organisms operated within this stable temperature system, contributing to continuous ecological interactions. The structure of the ecosystem where Titanoboa lived included interconnected habitats formed by rivers, swamps, and floodplains. These habitats supported diverse animal and plant life that interacted through feeding, movement, and nutrient cycling. Titanoboa existed as part of this system, influencing distribution and behavior of aquatic organisms through its feeding activity and movement across water environments. The mystery surrounding the disappearance of Titanoboa continues to attract scientific interest because it involves a complex set of environmental and ecological changes that took place after the period when this giant snake lived. Titanoboa existed during a time when Earth had very different natural conditions, especially in the region where its fossils were later found in what is now northern South America. After its existence, the environment began to shift in ways that affected many forms of life living in swamp forests, rivers, and surrounding land areas. These changes happened slowly across long stretches of time set, and they influenced temperature levels, water systems, plant growth patterns, and animal populations. Titanoboa depended on specific environmental stability, and any long-term change in those conditions affected its survival. One important factor connected to its disappearance involves changes in global temperature patterns after the warm period in which Titanoboa lived. During its time, the Earth experienced very warm climate conditions in tropical regions, and this warmth supported ecosystems filled with large reptiles and many water-based habitats. After this warm phase, gradual cooling began in many parts of the planet. This cooling affected the tropical regions where Titanoboa lived by changing humidity levels, rainfall patterns, and water temperatures. Rivers and swamps that once held steady warm conditions began to shift in structure and seasonal behavior. These environmental shifts reduced the stable conditions that supported the biological needs of large cold-blooded reptiles. Another element involved the transformation of forest environments. The land area where Titanoboa once lived was part of a broad system of wet and flooded forest environments. Over time, these forests began to develop into more structured and diverse ecosystems, with changes in plant types and forest density. The composition of vegetation influenced shade levels, soil moisture, and riverbank stability. These environmental adjustments affected the habitats where large reptiles depended on open swamp regions for movement and hunting. As forest systems evolved, open wetland spaces became less continuous, creating separated regions that altered how animals moved across the land. Water systems also played a major role in shaping the environment after Titanoboa's time. River paths shifted gradually due to sediment buildup, rainfall changes, and geological movement. These shifts affected the size and depth of wetlands. Areas that once held wide, shallow waters began to narrow or change direction. Such changes influenced breeding areas and hunting zones for large aquatic and semi-aquatic reptiles. The stability of water temperature and flow patterns also changed, affecting the availability of prey species that depended on specific aquatic conditions. These water system changes created new environmental pressures that affected survival patterns of large reptile species. Food availability also experienced changes during this time period. Titanoboa depended on a variety of aquatic and terrestrial animals that lived in swamp forests and river systems. When environmental conditions shifted, the distribution and population levels of these animals also changed. Some species reduced in number due to habitat loss or temperature variation, while others adapted to new ecological conditions or moved into different regions. This change in available food sources affected feeding behavior and survival stability. A reduction in consistent prey availability created stress on species that relied on large and steady food intake. Geological movement of Earth's surface also contributed to environmental change in the region. The landmass where Titanoboa lived experienced slow but continuous tectonic activity. This movement influenced elevation levels, drainage systems, and the formation of new land areas. As land elevation changed, water flow patterns shifted, and swamp systems began to reorganize. These geological processes took place over long periods and contributed to the restructuring of habitats. Changes in land elevation also influenced temperature and moisture distribution across the region, further affecting ecological balance. Atmospheric changes also played a part in shaping environmental conditions. The composition of gases in the atmosphere experienced gradual shifts over time, influencing rainfall patterns and temperature regulation. These atmospheric changes affected cloud formation and seasonal weather cycles. Rainfall distribution became less uniform in some regions, leading to periods of dryness in areas that previously maintained constant humidity. Such changes had direct effects on swamp ecosystems, which depended on consistent water input from rainfall. The reduction in stable humidity levels altered the structure of habitats that supported large reptiles. The reproductive patterns of Titanoboa may have been influenced by these environmental changes. Large reptiles often depend on stable conditions for egg development and survival of young. Temperature, humidity, and soil moisture all play important roles in successful reproduction. As environmental conditions shifted, nesting areas may have become less suitable for sustaining consistent reproductive success. Changes in seasonal cycles could have disrupted timing patterns related to egg-laying and hatching. These disruptions would have influenced population stability over long periods. Biological adaptation limits also contributed to the disappearance process. Titanoboa belonged to a group of cold-blooded reptiles that relied on external environmental heat to regulate body functions. As temperatures gradually changed, maintaining optimal biological activity became more challenging. Movement, digestion, and growth processes depend on consistent environmental warmth. Shifts in temperature patterns required gradual adaptation, and when changes occurred across long time spans, not all species were able to adjust effectively. This created pressure on population survival in changing environments. Ecological balance within swamp ecosystems also shifted during this period. Every ecosystem depends on interactions between predators, prey, vegetation, and environmental conditions. When one part of this system changes, other parts respond in connected ways. As plant types, water levels, and animal populations changed, the overall structure of the ecosystem began to reorganize. Large reptiles that once occupied important roles in these systems experienced altered conditions that affected movement, feeding, and habitat selection. These ecological adjustments influenced long-term survival patterns. Seasonal variation became more pronounced in some regions over time. Changes in rainfall cycles and temperature distribution led to differences between wet and dry periods. Swamp environments that previously maintained consistent water coverage began to experience more variation in water levels. This affected shelter availability and movement pathways for aquatic reptiles. Seasonal drying reduced access to certain areas, while heavy rainfall periods altered water flow patterns. These variations introduced additional environmental stress across long time periods. Disease and biological stress factors may also have contributed to population changes. Environmental shifts often influence the spread of microorganisms and parasites in ecosystems. Changes in temperature and humidity can affect the life cycles of organisms that interact with reptile populations. Stress from changing environments can reduce immune response efficiency in animals, making them more vulnerable to infections. Over extended periods, these biological pressures can influence population stability. Competition within ecosystems also shifted during environmental changes. As conditions evolved, other animal groups adapted to new habitats and resources. Some species expanded into areas previously occupied by large reptiles. These shifts influenced the availability of space and resources within swamp environments. Changes in species distribution altered interaction patterns within the ecosystem. These adjustments contributed to changing ecological dynamics across long time periods. Sediment buildup in river systems also affected habitat structure. Over time, rivers carry soil and organic material that gradually accumulates in certain areas. This process can alter water depth and flow direction. As sediment patterns changed, some wetland regions became shallower or changed shape. These physical changes influenced the distribution of aquatic environments. Large reptiles that depended on deep and stable water systems experienced shifts in available habitat areas due to these natural processes. Vegetation evolution continued to influence habitat conditions. Plant communities respond to environmental changes in temperature, rainfall, and soil composition. Over time, plant diversity increased in many regions, leading to more complex forest structures. This affected sunlight penetration, ground moisture levels, and riverbank stability. These environmental adjustments influenced movement pathways for large reptiles and changed the structure of swamp ecosystems. Long-term environmental cycles contributed to gradual transformation of ecosystems. Earth's climate naturally moves through phases of warming and cooling over extended periods. These cycles influence sea levels, rainfall distribution, and temperature patterns across regions. The period following Titanoboa's existence involved transitions that affected tropical environments. These transitions influenced multiple environmental systems at the same time, creating widespread ecological adjustments. Habitat fragmentation also developed as environmental conditions shifted. Continuous swamp forests became divided into smaller sections due to changes in water flow, land elevation, and vegetation growth. These divisions influenced movement routes for large reptiles and affected access to feeding and breeding areas. Fragmentation reduces connectivity between habitats, which influences population distribution and long-term survival stability. The disappearance of Titanoboa remains connected to a combination of environmental transformation processes that affected temperature stability, water systems, vegetation development, ecological balance, and biological adaptation capacity. These processes operated over long spans of time, gradually reshaping the ecosystems in which Titanoboa once lived.